we're gonna, as Olga said, kind of move from doing just kind of static quantitative proteomics into some of these more dynamic systems. So I'm really excited for this set of talks. If you didn't see on Monday, some quick disclosures. I'm currently a postdoc researcher at the University of Pennsylvania, but I also do have a company that I consult for called Talus Bioscience. So these days lately, I've really been interested in this idea of nuclear integrity. So what does that really mean? The nucleus inside of our cells is the house for our DNA, our genome, but it's also organized and read and written by proteins. So here I have two examples of two systems. I have cancer and I have aging. And just visually, it's very striking. And you can tell immediately that between healthy and these different disease states, the nucleus becomes very different. So here you can see in leukemia, the nucleus is typically quite small and compact, but then under the disease state in cancer, the nucleus becomes large, irregular, disorganized. When we look in aging, young nuclei are kind of smooth and compact, and then in older cells, in older people, the nucleus loses some of that integrity. And as the nucleus loses its integrity, this is really a function of the proteins that are trying to keep the nuclear membrane intact and keep the chromatin organized, losing their structure and losing their function. Now, nuclear proteins are actually a little bit difficult to study. So in our central dogma, we have DNA genes, which then get read by proteins. This turns transcription on, and then we get RNA expression, and then the protein is finally translated. There's really great measurement systems for studying chromatin biology like this. And for studying DNA, we have next generation sequencing, which has come such a long way in the last 20 years or so. We can use sequencing technology to do things like RNA-seq to measure RNA molecules. But if we want to actually measure the proteins that are organizing, scaffolding, reading, and writing DNA inside the nuclei, we really need to use something that reads the proteins themselves. So I, of course, use mass spectrometry for doing this. Mass spectrometry is super powerful. Hopefully you're all here because you don't need to be convinced of that, because we can make so many comprehensive measurements of the proteome. We take all of these mass spectra, we turn them into chromatograms, and then we're able to make these molecular maps, these phenotypes of what's happening to the proteome under different subtype conditions, cellular subtype conditions. Now, this is using protein abundance as kind of this static number, right? So here we just have all these samples, we have all the proteins, and we have how much we measured of them. But proteomes are so much more than just a sum of their parts. So later today, we're going to be hearing more from Isa about things like protein-protein interactions, the interactome of proteins. It's not just a single protein acting by itself in a cell. Typically, it's got its friends, its social network that it's going to be affecting structure and function for. There's protein localization. A protein that's in the cytosol versus a protein that's in the nucleus is going to be doing very different functions. And commonly, these are proteins that are going to be regulated, controlled by post-translational modifications. There's a lot of different types of proteoforms, and here I'm talking mostly about bottom-up proteomics, but different proteoforms and measuring them can be very dependent on specific sites, specific amino acids that are being modified, say by ubiquitin, by phosphorylation, or very commonly these days, people are starting to study glycosylation. And then we like to think of protein abundance as this single number. It's a quantity, but it's really not a static number. It's a snapshot of a dynamic number that's changing between proteins being synthesized and then proteins being degraded. So all of these today, we're going to talk about measuring them using various forms and applications of mass spectrometry. So I'm going to start talking about making measurements in quantitative mass spectrometry to start. So you've heard a lot this week about DIA, and we've heard from Brian Searle on Monday, and we've heard from Brendan McLean the past couple days. That quantitative DIA, there's a lot of methods, and the method that Brian and I use most commonly is this idea where we have our set of samples. We mix them together so that we have this library, this pool. We acquire all of our data. In the middle of our single shot DIA, we acquire 6 gas phase fractionated runs of that pool. So it's just multiple injection of that one pool 6 times. We build a library, a chromatogram library, using that data, commonly using predicted spectra from PROSIT these days. And then we search our samples for this library. So this kind of breaks down these steps into asking a question about what can I detect, and then asking a question of what I can quantify. So I want to dig a little deeper into what I can detect versus what I can quantify. So here I have a distribution of the yeast proteome. So this is showing in the log 10 protein copies per cell, all of the proteins along their abundance density. So this would mean that proteins on the left-hand side of this figure are low abundant proteins. Proteins on the right-hand side of this figure are high abundant proteins, and then somewhere in between is all of the other proteins. And most proteins that don't have extreme abundances, they fall somewhere in the middle here. If we were to measure this ground truth yeast proteome with mass spectrometry, we cannot detect everything. The things that are easiest for us to detect with mass spectrometry are these higher abundant proteins. Because they're higher abundant, they give us stronger signal in the mass spectrometer. We're more likely to make a detection. It's harder to detect proteins that are lower abundant. If we then ask the question, so this is what I can detect, we might want to say everything I detect I can quantify, right? But that's not the case. When we look at the peptides and the proteins that we can quantify, we see the exact same trend again, a subset of what we can detect. So this is showing overlaid in green. These are all the things we can quantify now. And you can see, just like detection, we can quantify very abundant things easily. But as proteins get less and less abundant, it becomes harder and harder to not only detect them, but also to quantify them. This is important because a lot of times the stuff that's low abundant is really important biologically. So here I'm just pointing out 2 particular proteins that have a lot of interest biologically, a TOR target of rapamycin protein and a sirtuin, SIR2. You can see that these proteins, very, very important biologically, fall on the bottom end, on the bottom half of this abundance profile. So these are proteins super important, but maybe hard for us to see. So this kind of paints a picture, this detection versus quantitation picture, that mass spectrometry is not inherently quantitative. When we measure things with mass spectrometry, we kind of assume that higher quantity will give us a higher signal. Lower quantity will give us a lower signal. And that signal, typically when we're doing DIA mass spectrometry, we're using an MS2 chromatogram. So if we say this small quantity, small signal, we have a small peak area. When it's a higher quantity, a higher signal, we should have a larger peak area. But at some point, even though the quantity decreases, there is no change in signal. So when we plot these quantity versus signal type charts, we kind of get a hockey stick shape. This is because every single analytical instrument, not just mass spectrometers, have this limit of detection, this limit of quantitation. At some point, even if you decrease the quantity that you're trying to measure in the mass spectrometer, you no longer get a change in signal. That point at which you've decreased the quantity, but there is no change in signal, that's a limit of quantitation. Why is this limit of quantitation important? Every single mass spectrometer and every single sample prep and every single peptide has its own unique LOQ, its own unique limit of quantitation. The lower the limit of quantitation, the better the biomarker is. This is because it means that that analyte, that peptide, that protein, that thing you're measuring can be measured more sensitively. You can get to a lower and lower quantity correlation for the signal that you're measuring on your instrument. What exactly does that mean in a proteomic standpoint? Here I'm taking one protein from yeast. This is a very abundant protein. It's a membrane-bound protein, so I've just marked out in gray where the transmembrane domains are. Similar to next-gen sequencing, we can make kind of a read pile. So here, all of the black boxes that are on top of this protein sequence represent a different peptide that we've detected with DIA mass spectrometry. So now we have all of these boxes stacked up. These are all the things we detected. But if we look at their calibration curves, look at their limit of quantitation, they're all very different. So here's one peptide that has a lot of variation in its signal. This is a peptide, there is a correlation of signal to quantity, but it is highly variable. It's very difficult to say for sure that some signal corresponds to a change in quantity versus just variation on the instrument itself. Compare that with another peptide, again, from the exact same protein. This peptide has a much smaller variation in its signal, so it's much easier for us to make the call that, yes, this change in signal corresponds to a change in quantity. We can pick a third peptide, all from this same protein, and this peptide even has some overlap with this other peptide. We look at this peptide now, and the variation is even smaller. So now this particular peptide, the signal can very easily be tied to a change in quantity. So this means 3 peptides, all from the same protein. One of them is not really quantitative at all. Another one is a decent biomarker, but not the best. And then finally, we have a peptide that would be the best peptide proxy for this protein. Now, this is just an example of one protein and 3 peptides in this protein. You can imagine every single peptide has this value. And it depends on the type of proteome you're measuring, on the way you prepared your samples, and on the mass spectrometer that you're using. This is not a static number that is the same for every single peptide, no matter what you're doing with it. So this means that now every single protein, every single proteome that we're trying to measure is a little bit different. So what we can do is build these kind of calibration curves. We can build them by picking whatever proteome we're interested in. So if we're interested in cerebrospinal fluid, we can pick an example of reference cerebrospinal fluid. You could actually just buy these from companies like Golden West. And then you can ask the question, what can I detect in cerebrospinal fluid? So we did this experiment, and we found for us in this healthy donor pool of cerebrospinal fluid, we detected a little over 1,000 proteins in that healthy donor pool. So now I've just emphasized multiple times, not everything you can detect is quantitative. When we look at the calibration curves for all of those peptides we detected, we find that there are about 666 good biomarker candidates. So this is things that had any change in signal when we change the quantity using these calibration curves. Now, typically to do this step would take weeks to months, because every single one of those proteins that you're interested in, you would order stable isotope labeled standards for each one of the peptides you want to assess, and then build the calibration curve, measure the data, and analyze LODs and LOQs. So next, you might want to do the highest quality preclinical biomarkers out of this. So now you're doing more like calibration and validation tests that require those heavy isotope standards. And as I mentioned, every single one that you want to purchase is going to cost you about $1,000 per peptide if you're doing a heavy isotope standard for them. So this process using kind of conventional methods is going to take you weeks to months just to decide which biomarkers you want to chase after from your system. Then it's going to cost you about $1,000 per peptide that you want to test for preclinical validations. If we use DIA, now we can measure every single protein, all of the peptides in the sample. If we use this approach that I had published last year called matched matrix calibration curves, now instead of spending weeks to months to figure out what you can detect in your sample, it takes you 2 days. You do one of those gas phase fractionated libraries, you figure out what you can detect in your sample, and then instead of spending $1,000 per peptide for stable isotope labeled paired standards, you spend about $1,000 per proteome using either SILAC to shift the mass MZ values of your peptides, or you can use other methods to dilute your reference proteome in a background. So DIA has already really enabled a lot of advances in doing quantifications and doing them very quickly. So I'm going to change gears in just a minute, but I could also take some questions. I see there are already some questions in the chat.
Yeah, there's one question which may be relevant now. So from the proteomics point of view, could you please kindly summarize what is a good biomarker?
Yeah, so proteomics biomarker, there's a couple good papers, and if one isn't able to get to them in the chat, the two good papers as far as biomarker validations, the first one is Andy Hoofnagel and Russ Grant. The title of it is something like Paradise Found or Paradise Lost and Found. It's about a 3-page paper, very quick read, that highlights all of the experiments you should do to get to this kind of preclinical biomarker stage. Again, that's not like a validated assay to put in patients, but it gets you to the preclinical stage. The other paper, senior author, last author is Steve Carr, and it's the Fit for Purpose paper. So these two papers are kind of, I would say, like the gold standards for doing preclinical biomarker discovery in proteomics. There's further clinical validations. CLSI has a workbook, but that's like super thick of all like kind of the FDA regulations to get to an actual clinical biomarker. So it's a great question. I think we got that one. Question from Ahmed, how did we determine the LOQs of peptides? That's in this paper, which we're not going to like dig super into the details, but this J Proteome research paper from last year goes over the methodology for building the actual samples for calibration curves. And then it also has link to the data analysis to calculate LOQs from your data. You can also calculate LOQs using MS Stats. I'm not sure if you're going to learn that later this week, but once you learn how to use MS Stats, it's very easy to use this function in MS Stats to calculate LOQs. I'll start at the top. Raja has a question. Protein copies per cell is being done using histone proteomic ruler and DDA LFQ. So the protein copies per cell that I had on those first slides, that's taken from a paper by Sina Gamagami. Great proteomics work, but this was like a Herculean effort in yeast where every single ORF, every single open reading frame in yeast was tagged, and they did like actual counting to get those protein copies per cell. So we use that as the ruler in that case. Is there a good agreement in copies per cell between DDA and DIA? I was not going to talk at all about comparing DDA and DIA, but there are some great papers. I'll see if I can dig them out that are comparing that. A little bit later, we're going to do some comparisons of DDA versus DIA quantification. DDA is good for like a limited dynamic range, but DIA has a much broader dynamic range. So if each individual peptide has its own variation, is this taken into account during quantification in the case of shotgun proteomics? As far as I am aware, there is no algorithm out there that really takes into account that different peptides have different LOQs. I think most protein quantification, you'll be learning more about this later too, I think most protein quantification, I think the most common way people do it is literally just summing peptides to get a protein level quant. Like a top 1 or a top 3 or a top n, just summing everything up or taking the average. There are some more intelligent ways of doing this that MS Stats takes into consideration, but that does not include like specific LOQ values. Coco, we will post names of papers. If the TAs don't get to it, remind me in the chat and I'll get links for later. And these slides will be up after the talk today. So you'll have the DOIs for that. Okay, so I'm going to move on for now. I think we got mostly everything. And maybe I might get back to some of these questions later. Yeah, so in the context of biology, I had mentioned earlier how now we have these giant maps of looking at differential abundance as a molecular phenotype for different perturbations, different molecular subtypes. So one question that I had asked previously was whether there was a different molecular phenotype for lifespan extension. So here I was using yeast, so everything was being done in this yeast model organism system. But what I wanted to ask is, can we extend lifespan? Can we have yeast that live longer, either through genetic mutations or through environmental interventions? And I kind of expected I was going to see this phenotype reflected in protein abundance. And that's because there had been previously this paper where they had measured yeast lifespan by deleting every single gene in yeast. So another Herculean effort, they deleted every single gene in yeast systematically and measured the lifespan of the yeast. And they found that there were some pathways that extended lifespan. These yeast lived longer. There were kind of major themes here when they deleted each of these single genes, which deletes the protein. And they found that a couple of these pathways were things that to me really spoke to the protein level and the proteome itself. Things like ribosomes, so that spoke to me saying the synthesis of proteins is being affected. The proteasome, the degradation of proteins is being affected. Saga is a protein complex in the nucleus that kind of can regulate different gene expression. So kind of all of these taken together, I was thinking there must be something in the proteome then that is driving this phenotype of extended lifespan. Let's look at protein abundance and see if we can find it. So I picked a couple of these single gene deletion yeast strains and I wanted to grow them up in bulk, measure their proteomes with DIA, and I was going to see this beautiful signature in the proteome. I did what's called a set cover approach. And what this set cover does is, given two sets of information, I gave it the functional overrepresentations, and I gave it this protein interaction network because I wanted to optimize for a couple things. I wanted to optimize for single gene deletions that extended lifespan across a range of values. So I didn't want all very large lifespan extensions. I wanted to get a range of lifespan extension. And I wanted to get a number of unique genetic interactions. So I didn't want to do all gene deletions that were ribosome gene deletions. I didn't want to do all that were minosyl-transferase deletions. I wanted to kind of cover as much as possible across the functional overrepresentations and have the most unique genetic interactors, the most unique protein-protein interactions. I ended up with this list, which surprisingly included two of the Saga complex members, which was interesting in itself, but seemed to span the range. None of them were slow growing. They had a lot of different genetic interactors. So I thought for sure I'm going to see like massive changes in the proteome when I compare these single gene deletions to regular yeast. You can probably tell by my slightly sarcastic tone, that's not at all what we saw. So when we delete TOR out of yeast and measure the change in protein abundances between that TOR deleted yeast and the normal parental genotype yeast, really not a lot is changing here. A couple of these gene deletions are actually just artifacts in the genetic makeup of the yeast itself when they made these single gene deletion strains. I look at a couple of the others, and again, I really am not seeing huge changes in the proteome. The biggest change that I ever saw was this SGF73. So this was really not promising for my hypothesis of this molecular phenotype of lifespan extension in yeast when I looked at each of the single gene deletions. So it was not surprising when I tried to say, is there a proteomic signature of lifespan extension in yeast? There really was not. There's really nothing here. Even when I compare lifespan shortening, so shorter lifespan yeast, there's really nothing in the proteome here. So this was really surprising to me because I kind of thought like, obviously this has to be a change in the proteome. There has to be a proteomic signature because what other molecule is going to be affecting the phenotype? It's not going to be the DNA that just kind of sits there. It's not going to be RNA. RNA doesn't really do anything. It's got to be the proteins, right? Didn't see that at least in the abundances. So I kind of put that disappointing result to the side and I said, okay, it's not single gene deletions. What about environmental interventions? So it turns out actually you can put yeast on a diet just like a person, just by changing how much glucose it has available. So at these higher glucose levels, calories are abundant, and the yeast has a phenotypically normal lifespan. At some point, when you decrease the glucose too far, when you limit its calories too much, the lifespan goes down, kind of starvation type of thing. There's a sweet spot in between, however, where the glucose is present but not overly abundant, where lifespan extends. There is a point of calorie restriction where lifespan increases on yeast. And I said, okay, this is going to be it. This is where I'm going to see my proteome changes. And I kind of hypothesized across a range of glucose availability, I might see that reflected in the proteome a couple different ways. So the first would be across availability, the peptide abundance, the protein abundance would not change at all. These are proteins that are not affected at all by the amount of glucose. I might see a dose-dependent change in protein abundance, where maybe a particular protein is highly abundant when glucose is available, and then lowly abundant when there isn't glucose, or vice versa, low abundant in high glucose, and then becomes high abundant when we move to low glucose. When I looked at my data, I didn't really see either of those trends. So each one of the lines here represents another peptide, and I'm plotting those lines across the different glucose availabilities. So I didn't really see either of those trends. Mostly it kind of looks like flat lines. But one thing that I did notice when we were looking at this was some proteins seem to almost have a signature where they're high expressed in high glucose and then low expressed in low glucose, kind of like a switch. So it's abundant at the same level in one condition and then abundant at a different level under a different condition. So when I reanalyze my data that way, where I group the high glucose and then the low glucose and compare those proteomes against each other, I see a huge change in the proteome abundances. This was more what I expected to see when I was deleting genes out of yeast. So comparing the low glucose to the high glucose, there's a lot of protein changes here. And this is something you can do. I just made these figures directly in Skyline using the group comparisons. So if it looks familiar, that's why. When I look at some of these functional groups that are being enriched or depleted between the glucose conditions, it kind of makes sense. So under limited glucose, things like the ribosome biogenesis are being downregulated. Under low glucose, things like carbohydrate metabolism are being up-regulated, increased abundance. So these kind of make sense in the context of the glucose level, but did it make sense in the context of lifespan extension in yeast? So some of these proteins that we did see with increased abundance in that low glucose sample are documented, known as having lifespan extension properties under calorie restriction conditions. So this is one protein, a heat shot protein, HSP12, and I'm showing the peptide trace from in Skyline, where you can see for this protein, Skyline plots each one of the peptides for that protein that is measured for that protein. So each line is another peptide from this protein, and then each one of the points along the horizontal axis is another one of those samples. So you can see this protein, all of the peptides are low, low, low in these first three glucose conditions, and then all of the peptides very nicely, stably become increased abundance once we hit somewhere between 0.5% glucose and 0.05% glucose. At some point in there, this gene is turned on, transcription is turned on, and the protein becomes abundant. So that was kind of interesting. It definitely didn't prove my hypothesis right at all. My hypothesis was definitely proven wrong. And I kind of took away from this 3 lessons. So the first lesson I learned was to my surprise, just deleting a gene might affect a phenotype, but it didn't seem to affect the protein abundance. The proteome seemed pretty plastic, pretty stable in response to deleting an entire gene, which was not what I expected. I found that although genotype manipulations didn't seem to change the proteome, calorie restriction really did. So environmental modulations had a much stronger effect on the proteome. And then finally, what I really took away from this is that protein abundance alone doesn't necessarily mean there's a mechanism of action. So there has to be a way to get beyond the protein abundance and ask questions about the functional proteomics. So you might tell we're getting ready to switch gears. So I'll stop again for questions, but it looks like the TIAs have done a pretty good job. There was a question about the low glucose conditions. They were exposed to low glucose for roughly 24 hours. I tried to control for growth rate and harvest so that the population doublings were about the same for each. So the time was a little different for each of them. Yeah. So we arrive at this message: proteomes are more than the sum of their parts. Protein abundance is great, but isn't always the full story. Sometimes you see no changes in the proteome, but there's clearly something happening molecularly in these cells. So I had kind of alluded to this, but the next logical place to start looking, to start exploring, is in protein turnover. So I mentioned how protein abundance is thought and represented as this static number, but it's actually a very dynamic value that we're only taking a snapshot of. So the protein abundance will increase if you're synthesizing more protein or if you're degrading it less. And the abundance will decrease if you're degrading it more or synthesizing it less. And what we see when we're measuring a protein abundance is just at that moment in time, the snapshot of these two molecular processes. So using mass spectrometry, using this technique, pulse SILAC, can capture both of these pieces of information. So in a pulse SILAC experiment, you would take your cell culture. For me, I was working in HFF1 mammalian cell line, and you grow your cells in regular media to start. Once the cells are happy and proliferating, you switch the cells to a heavy media. So this is media that you create where you purchase heavy amino acids. You purchase media that is depleted for those amino acids, and then you supplement your media with these heavy isotope labeled amino acids, typically lysine and arginine. So now, as time passes, as your cells are growing, as they're synthesizing proteins, you are the cells are including these heavy isotope labeled amino acids to their proteomes. So if you look at a ratio of the light to heavy proteins in these cells at time 0, where they've only seen light media, all of their proteins contain 100% asterisk. It's not entirely 100%. It's naturally abundant isotopes, but 100% light amino acids in their proteomes. And then as time goes by, they start incorporating more and more, synthesizing more and more heavy proteins that contain these heavy amino acids. At this point, this 50-50 point, it's called the protein half-life, so the turnover value. So when I look at this as a quantitative proteomicist, I notice two things. The first is that all of these measurements start at 0. So this curve here, to get to the 50%, to shape this curve, having these initial points is very important. Having these low abundance ratios is very important. The next thing I notice is that these protein synthesis values occur on the order of hours to days. Some proteins are extremely quick and are turned over very quickly. Some proteins are very stable and stick around for days. So these are kind of two aspects that suggest we need a very sensitive ability to measure dynamic range. So we need dynamic range in the time dimension. We need dynamic range in this ratio measurement dimension.
Lindsay, there's a technical question. Are these time scales, for example, in terms of hours, typical of protein turnover?
Are these time scales typical of protein turnover? Yes. Yeah. So these time scales between like hours to days is typical of all kind of protein expression. Every protein is a little different. And honestly, every proteoform is a little different. So we'll talk a little bit about getting at that proteoform question in just a bit.
And another
Yes.
question related to this. So 50-50 light and heavy, shouldn't the ratio of light versus heavy be 1?
Yeah, sorry, this label that I gave it here should be over total. Sorry about that. That was my fault. I'll definitely fix that slide before I post it. Have I seen protein turnover in 30 minutes? So when you model these, it really depends whether you see turnover in 30 minutes will really depend on how many time points you collect really quickly there. There is a full range is what I will say. Susanna question, will the curve be slightly different for every protein? Absolutely. Every protein will have a slightly different curve here. It's a protein dependent process. I will let one of the TAs grab Ahmed's question because that is something I think that they can answer. And I think maybe BK's question can be answered by the TAs too. So I'll keep going. So I think kind of the logical thing that everyone's thinking, considering this is a DIA focused workshop, the question becomes naturally, can we do pulse SILAC and measure it with DIA? One of the first questions is, how do you analyze that? How do you process this data? So I just described kind of this protocol of how you would start cells in a light media and then grow them into a heavy media. For these experiments, I did the exact same style of gas phase fractionated DIA library, the chromatogram library approach that I use in my other experiments. Here, after you're done collecting your data, you have to do a bunch of post-processing in order to calculate these half-life values. But kind of this general process, you do your pulse SILAC experiment, you acquire your data, and then you calculate these protein half-lives. Just to give a little bit more detail on this vague acquired DIA MS data, what we thought to do is for this chromatogram library, because we're doing pulse SILAC, we're really only interested in the longitudinal changes in abundance after time 0. So what we can do is we can generate a time 0 chromatogram library. And just like Brian had talked about, I like to use ProCit to predict my spectral library. And then I use DIA analysis and gas phase fractionated DIA analysis using Encyclopedia. And now I've built this time 0 library. Then using that time 0 library, again in Encyclopedia, I detect all the peptides in all of my samples throughout the pulse SILAC time course. And what that does is it performs a retention time alignment for me. So now I have all of the peptides that are detected and the retention times aligned. And now I can use Skyline to pull in those chromatograms for the light and the heavy paired proteins. This is very quick to do in Skyline. You guys are all becoming Skyline experts now. But what we can do, we just add all of the peptides to our target list using that library from Encyclopedia. And then you can very quickly, in about 3 clicks, add the heavy isotope labeled standard version of all of those peptides. So now I have this workflow using all this free open source software that you've heard about this week. The question becomes, how does it compare to DDA? So we wanted to benchmark this. Sounds like a great idea. In theory, maybe it'll work. We're not sure. So we took a heavy proteome and a light proteome, and we mixed them together in ratios. This is kind of from the Navarro paper some years ago. This is kind of the idea of that hybrid proteome. So now we have known ratios that we can compare to our measured ratios. So if we do a conventional pulse SILAC, this is pulse SILAC DDA, we see that the measured ratios, those are each one of the colored points, the measured ratios, and we can compare them to the expected ratio. That's these horizontal lines. The N up here just indicates how many detections that we made. So out of all of those detections, we can visualize this with a box plot to compare the measured ratio and the actual ratio, the expected ratio. We can see for some of these less extreme ratios, DDA, conventional SILAC, does a pretty good job of recapturing the expected ratio. So you can tell that these box plots are kind of small, and the mean is right about at that expected ratio line. But once we get down to about a 1% ratio, so that's 1 to 99, the measured ratio has started to kind of already hit its floor. So we kind of get this leveling out of the measured ratio once we get down below this value. We can compare the exact same samples measured with DIA using that pulse SILAC approach. And one of the first things that we spot visually is that the abundance range, the dynamic range of the measured abundance is much wider when we use DIA. DIA is being done on the MS2 level. So we get a wider dynamic range in a measured abundances, but we also gain an order of magnitude in quantitative accuracy. So that order of magnitude is this light blue box is now jumped down more closely to the expected 1% ratio than if we can compare the conventional pulse SILAC samples. So we wanted to apply this in maybe a more interesting system than this benchmark. And what we decided to do was look at bortezomib treatment. So this experiment is performed the exact same way. So we're taking these human cells, growing them in light for a while. Then we're going to switch them to heavy media. But at the point when we switch them to heavy media, we're going to treat one set of samples with DMSO. The other set of samples we're going to treat with this drug, bortezomib. So bortezomib is a proteasome inhibitor. It's the first FDA approved, first in class proteasome inhibitor used primarily for myeloma indications. This proteasome inhibitor, what we expect is proteins that normally would have been degraded by the ubiquitin proteasome pathway are now no longer going to be degraded. So they're going to be around longer. They're going to have longer half-lives, longer turnover values.
Lindsay, a question related to that. Why do we use DMSO as a control in this type of studies? Is it truly inert?
Yeah,
Why not just add a smaller amount of millicube water?
Yeah, that's like an excellent med chem question. DMSO is just kind of the standard. I actually don't know historically why we don't use water, but definitely in med chem, everybody uses DMSO for their control. Yeah, usually Leah has a good point in the chat. Usually when you're bringing up these drugs, you get the drugs as like a lyophilized like powder, and typically you resuspend them in DMSO, not in water. Yeah, but I'm not sure why we, like science as a field, originally picked DMSO to reconstitute these versus something else.
Yeah, another comment by Raja. Yeah,
sorry, just to mention, I think it's because DMSO can actually dissolve way more things that you could in just an aqueous solution.
Yeah, there's definitely the argument for like using water is only going to get water soluble things, and some drugs are not easily water soluble. So yeah, I guess while I'm looking at the chat, I do not personally have a good resource for examples of protein turnover timescales, but if someone knows one off the top of their head, please let us know and we can hook that up. How does pulse SILAC DIA compare to pulse SILAC TMT in terms of accuracy and precision? I have not seen a paper comparing that with pulse SILAC, but Professor Vitek, Olga, actually has a good paper with, I think it's Lucas Reiter, that compares DIA with DDA. Maybe someone can drop a paper for that. I don't know it off the top of my head.
I will add the reference in the Google Doc.
Perfect. Yeah, but that's an excellent paper. Kind of one of the take-home messages I got from that paper comparing DIA with TMT is that they are about the same in terms of accuracy and precision. And it's kind of just whichever one you're more comfortable doing. Like, for example, if I tried to compare DIA and TMT, TMT would probably look awful just because I've never done it before, but I've done DIA a lot. I'm sure there's other labs out there that are very, very comfortable doing TMT, where their DIA might not look as good as their TMT.
That's pretty much the general conclusion.
Perfect. Glad my too long don't read version of the paper hits the right take home message. So I haven't seen it in Pulse Silac. There are additional confounders with Pulse Silac that maybe that would change. Just kind of like the isotope distribution of the Silac on top of the isotope distribution, or I guess not isotope because that's MS3, but the TMT takes advantage of isotopes. The Pulse Silac takes advantage of isotopes. It might work out because it's kind of different isotopes, but at the same time, it might cause some interferences. It's like a really good question. Good thought experiment. You could probably do some good in silico simulations to see if that would work. Okay, I think we're caught up with questions. I'll just share with you some of these results. This is what those turnover curves look like. So this is showing the time as it progressed along the horizontal axis, and then on the vertical axis, it's the fraction of the light to the total protein abundance. So when we start out at time 0, all of the proteins are 100% light. But you can see even just by eye looking at these curves that some proteins really are not being turned over at all. They remain 100% light pretty much forever. So those are proteins that are just not being synthesized more, and the light is not really being degraded. The light at time point 0 accounts for pretty much all the protein that's there forever. And we know this is the case, right? Because like there's proteins in your eyes that you're born with that you never get new proteins in your eyes. That's it. So we know that this is the case for some proteins. You can see the majority of proteins turn over at like kind of a moderate rate, about maybe like 150 to 100 hours. But we can calculate these actual values by calculating the 50% point, the half-life in these curves. And when we do that, we can make a pseudo volcano plot out of the differential proteins. So this is not a real volcano plot. So I'm going to kind of explain it a little bit. So here, instead of a log2 fold change, we put the log2 of the turnover value for bortezomib over the DMSO. So if there's not really any change, it's like a volcano plot, not really going to be interesting. The larger the log2 value of this ratio gets, the more different the turnovers are. And then we get these p-values. So if we look at the statistically significant differences in turnover, we get this handful of proteins that are being differentially turned over under bortezomib treatment. If we look up some of these proteins, they cover a range of protein half-life values. Real quick, I'm going to point out in red, these 3 proteins are known. They're documented as being part of the ubiquitin proteasome pathway. So that's reassuring that we're recapturing some things that we know about the ubiquitin proteasome pathway. One thing that was interesting to me is these 3 proteins that actually have an opposite half-life. They're not being extended, they're being turned over faster, it seems. So these are kind of interesting things to see. It kind of makes me wonder, you know, this decrease in the cathepsin D, this CAT-D protein, the second one here, could it be explained by, one, it could be an increased synthesis rate. Cathepsin D is an aspartyl protease, so it's kind of maybe orthogonal to the ubiquitin proteasome pathway as a protease itself. Maybe cells are trying to upregulate this protease activity to try to clear out some of these accumulating proteins that aren't being degraded normally. Cathepsin D, this is also a precursor. Is this a proteoform that we're not capturing because we're just doing kind of regular bottom-up quantitative proteomics? So could this precursor just have another form, another isoform that we're not really capturing well here? So this idea of even when we break down protein abundance into its component synthesis and degradation rates, there are still things functionally that we can't quite explain at the proteoform, at the functional level, kind of led me to some of my next work. So it looks like we might be doing okay for questions. So I'll keep going real quick. So I mentioned in this last work, looking at protein turnover, we were looking at bortezomib, which inhibits the proteasome and the ubiquitin proteasome pathway. So logically, that kind of led to my next interest, which was looking at post-translational modifications, ubiquitin. So in a lot of my systems, what I'm interested in is processes like aging or like cancer, where the DNA itself might not change at all, but the proteome, what's driving phenotypic differences. And in my postdoc at the Garcia lab, it's not too surprising, the main PTM we're interested in is histone tails. So in the context of aging, as you age, there's a lot of changes going on in your nucleus. So inside the nucleus, under normal chromatin states, as we age, we start to accumulate different protein variants in our chromatin. There's certain histone proteins that become incorporated more and more with your DNA as you age, as cells age. There's different histone modification patterns. The DNA itself might become mutated, and the DNA itself might be modified differentially. So measuring histones themselves, there's really only like the 4 canonical histone proteins that make up a nucleosome. But when you compare the complexity of those 4 proteins and all their possible PTMs, the problem becomes very large. For context, out of like the 40,000 human proteins, there's about 1E8 peptides to search for the proteome. There's only 4 histone proteins, but when you take into account all of the combinatoric PTMs that are possible on them, this becomes as hard a problem as comparing the size of an Egyptian pyramid to the size of the entire Earth. So it's a very difficult problem. So since we're kind of talking in the context of Skyline, I want to highlight some of the difficulties, not just in this search space, but also in Skyline, even when you're looking at these proteins. So here's one protein. This is histone 4. It is a mess. It is an absolute mess. So histone 4 can have a number of different residues: lysine 5, lysine 8, lysine 12, and lysine 16. These 4 residues, any one of them can be acetylated. So this is an example of that combinatoric problem. So each one of those residues, one of them could be acetylated. The others can be unmodified. 2 of them can be acetylated, or 3 of them can be acetylated, and then finally all 4 of them can be acetylated. So this is one peptide from one protein and just one type of PTM. There are other types of PTMs that happen on histones. So not only is that difficult just from a search space problem, but I'm showing you their chromatograms, and I'm showing you that for each type of modification, whether it's a single acetyl, double acetyl, or a triple acetyl, chromatographically, they do not resolve. So now we get all of these peptides co-eluting on top of each other, and they all have the exact same mass to charge value because they all have the exact same sequence and the same number of modifications. So their precursor mass to charge is the exact same. So this is just one example of one peptide on one protein that makes histones so tough. There is also, I mentioned how when we age, there are different variants of proteins, of histone proteins being incorporated into our chromatin. So here I'm comparing histone H3.1, so this is one histone H3 variant, with histone H3.3, another variant. There's a single amino acid change, an alanine to a serine. So when we look at this peptide sequence in one protein versus another protein, there are 3 peaks here. Which one is correct? There's a slight mass to charge shift, but which one of these peaks is the right one? If we're using DIA, unless we have a large enough MZ shift that it falls in a different DIA window, a different DIA isolation window, we are co-fragmenting these. So which one of these peaks corresponds to the right peptide? So histones, all of this to say, are very, very difficult to analyze. Some of the work that we've done is using Skyline and using this idea of the prior information, using things like spectral libraries, using things like IRT calculators to process our data and look at our data, so that we can be more convinced that what we're measuring and what we're reporting is correct. Oh, there was a quick question in here about derivatizing the lysines. All of this that I showed you, this is all with the lysines derivatized. This I say it's unmodified, but it's actually propionylated. So these are all derivatized, and chromatographically that does not change their elution off of C18 column at all, which is unfortunate. Okay. So we're going to use Skyline. We're going to use as many tools as possible in Skyline to make our life a little bit easier. So I put together kind of this collection of scripts. They're definitely still in a work in progress stage. But what this collection of script is going to do is first, it's going to help you build a Skyline document. So this is especially useful if you're doing some of these histone mutants. There's also oncogenic histone mutations, point mutations in histones. This allows you in Skyline, you've seen how easy it is to add any new target you want. You can simply type in a sequence you're interested in. You can add new modifications. So commonly for histone PTMs, people are interested in kind of the usual suspects, things like methylation, acetylation, but there's growing interest in other types of modifications like glycosylation, like citrullination. Using Skyline, it's very easy to add modifications just by right-clicking on the target and going down to that modifications option in the drop-down menu. And then finally, doing non-model organisms. So I have collaborators that are honestly doing all kinds of organisms, from slime mold to leafcutter ants to, of course, human. So all of these are going to require a slightly different approach. Using Skyline, it's very easy to accommodate any of these experimental systems. Next, we're going to use DIA to acquire this data. And I'll talk a little bit about how we have to think about modifying a DIA method for different types of systems. Brian touched on this a little bit when he mentioned how if you're doing phosphoproteome and you want to do DIA, adding that PTM changes a little bit how you should set up your method settings on your instrument. And then finally, a lot of these analyses are going to be done using Skyline, but accompanied by some Python scripts that I've been working on just to make this a little bit easier. So the very first thing is just building that target list in Skyline. So the target list in Skyline on the right, just a snapshot here. We've all seen how you can add peptides and proteins, but one of the first scripts that I wrote was to take a list of modifications that you're interested in. So this has lysine 4 acetyl, lysine 9 trimethyl, serine 150 phospho. Take a FASTA of the proteins you're interested in, and then using what's called a Cartesian product, calculate all the possible combinatorics for those peptides. So it's important in Skyline that we're kind of monitoring for all these combinatorics to be sure that what we're seeing, the peaks, the chromatograms that we're seeing, are truly belong to that particular isoform, that particular PTM peptide. So now we have a target list. One of the other tools that we're using in Skyline is the IRT calculator. So when we prep histones, first we isolate histone proteins from the nucleus. But that isolation is not perfect. When we're enriching for histones, a lot of times we get other proteins that are co-enriched with those nucleosomes. We can use those co-enriched peptides to build an IRT calculator. So we don't have to use Biognosis. We don't have to use commercial kits to spike into our samples. We can make our own calculator from common contaminants in the sample, or here a lot of these peptides are ribosomes, not things we're interested in from an experimental standpoint, but things that we're going to be measuring anyway. And just as a reference in here, I'm not going to walk through this entirely, but building these calculators is very straightforward. I'll add this as a reference so you can kind of play with it yourself. But just when you get to this edit IRT calculator, in the drop-down menu, you can choose add, and then you can paste whatever peptides you want to use in here. There are some tips and tricks that we can talk about if people are curious to pick peptides that are going to work well. But the sky's the limit. Anything that's in your sample, you can build an IRT calculator for. Okay, so we're built. The Skyline document is ready. The next step is to acquire data. So acquiring the data in DIA, we've now looked at DIA data a lot of different ways. This is going to be yet another way to look at DIA data. So here I have the chromatography on the horizontal axis. So this is the measured retention time for the histone PTMs in kind of like a standard PTM analysis. Then on the vertical axis, I have the precursor mass to charge of these histone PTMs, and I am binning them by the isolation window scheme. So using a previous method in the lab, we used 50 MZ wide windows, 50 mass to charge wide windows. So as we look at this one slice of retention time, some of these windows have 0 precursors. The next window up, 350 to 400 MZ, has 1 precursor in it. If we move farther up, some of these windows have 3 precursors in them. These are 3 peptides, 3 precursor ions are being co-fragmented in the window. If we expand that out, this is what that whole data set looks like using a 2015 method published from the lab. So this is not maybe an ideal DIA method using some of the things that we've kind of mentioned along the way so far today. So an ideal method for PTMs would not have co-fragmented isomers. It would have a fast cycle time. You would get enough of those points across the peak that Brian talked about. And then finally, it would be nice if we didn't have idle windows. We're not spending time collecting data in windows where there's nothing we're interested in. So a couple of things that we can do to optimize this method. We went from a 16 MZ, 16 windows of 50 MZ precursor range to a 25 windows of 24 MZ using that staggered approach that Brian mentioned earlier. So now we have 12 MZ effective windows. A couple things in this DIA method optimization. The first is we reduce the precursor range. Instead of acquiring the 900 to 1100 MZ range, up here, a lot of these peptides, a lot of these precursors are actually just lower charge states of things that are down in this busier section of the method. So we'll cut out these lower charge states and focus on these higher charge states that are down in the range of 300 to 900. The next thing we did by decreasing those windows and using that staggered approach, as Brian had mentioned, we kind of get twice the speed for nothing. We're getting reduced window size using these staggered methods. So we went from 50 MZ wide windows down to 12 MZ wide windows. And what does this mean? So let's take a look at just zooming in on a couple parts of the gradient. So this part of the gradient at this particular retention time in these 2 windows, we are co-fragmenting 4 precursors because these windows are so wide. When we decrease the size of the window, now we have separated 1 of those precursors from the 3. So we didn't do perfect. There are still 3 precursors falling in some of these windows, and those 3 precursors are within 12 MZ of each other. We can see other points of the gradient where we had 2 precursors that co-elute at the same mass to charge value, co-fragmenting in this window, but they don't have the exact same precursor value. So if we lower the size of the window, we decrease the size of the precursor window, now we've separated out these 2 co-eluting isomers. Because they co-elute, but they fall in different windows, now we can more easily pick them apart in Skyline. So I'm seeing a lot of questions about what kind of MS. Here's a reference chart for you, and these slides will be up. So here's some proposed methodologies that you can use, just compared to kind of what the previous standard was. The 3 in the middle are Orbitrap instruments. And this last one is from a paper from the Dannon's lab doing swath, so on a triple TOF. I haven't personally been on a triple TOF yet. Someday I'd love to get on it, but these slides will be up later if you wanted to play around with this, these data acquisitions. But the last thing I'm going to talk about, analyzing the data. So now we've built our Skyline document, we've acquired our data, we've pulled it into Skyline. It's still not over. now it's the hard part. That was the easy part. So now it's the hard part. Why is it hard? I talked about these co-eluting, co-fragmented isomers. So here's another histone protein. This is histone H2. Lysine 4 and lysine 7 can be acetylated. Monoacetylated. The rest of these, the question before about the derivatization, the rest of these lysines are derivatized, are propionylated. So these two precursors, these two peptides, pretty much coelute. When we look at the fragment level, many of their fragments in the fragment letter are going to be shared. So in the Y series of ions, all of these ions down here are all going to be the exact same between these two peptides. What we should do to perform good quantification, to be confident in our quantification, is only use the fragments that are unique to each of these peptides, site localizing. In PTM, we talk a lot about site localizing fragments. So let's only include fragments that include the PTM modified residue. So when we do that in Skyline, we can mark these specific transitions, which are unique. And what that looks like is the dotted lines are not unique. Those are transitions that are shared between these two peptides. And then the solid lines are the ones that Skyline is going to use for quantification of peak areas. It'll still use the other fragments to perform detection, like peak detection, but it'll only use the solid lines for quantification. And that's very straightforward to do in Skyline. You can either right click on the target itself, or you can open up the document grid, go down to the transition report, and then it'll show you all the transitions in here, and it'll give you a checkbox to say if they're quantitative or not. And I can link you guys to some tutorials that are still work in progress. They're not published or anything yet. You can very quickly, just in Excel, concatenate the precursor to the product MZ, and it'll give you a true false for whether or not it's quantitative. So now we can have this document, and you'll see that we can have in our document grid, we can check the things that are quantitative. We can look now at the precursor. The precursor is not quantitative because there's a lot of isoforms for this particular peptide. And when we look at the different fragment ions, we could see some of the fragment ions that are quantitative versus not quantitative. And then Skyline will not use the ions that are marked not quantitative. There's a lot of other helper scripts out there that you can look at. So this is that paper from the Dannon's lab that I mentioned before. But everything up until this point assumes that you're not doing anything else. You're only doing DIA for your data acquisition, and then you're going to analyze it in a targeted, like PRM style manner. If you wanted to discover new histone PTMs, you could do this kind of approach where you're pairing DDA with your DIA. And then you build a spectral library using whatever database search is your favorite. There's a couple that are better for histone PTMs specifically. But whatever database you like using, you can build your spectral library in Skyline using that bibliospec built-in tool. And then once you have your DDA library set up in Skyline, you have all the MS IDs in your document to pull in the extracted ion chromatograms of your DIA data using the detections in that DDA. So that ends up looking something like this. So when you have one of these paired DDA spectral libraries in Skyline, you can visualize each one of the MS IDs in that DDA search on top of your chromatogram, and it makes these vertical cyan colored lines. So you can see this chromatogram that has been detected in Skyline. It's picking the peak right here where we've got all of those paired DDA MS IDs. So this is a really powerful approach. We just spent so much time talking about not using DDA for our DIA analyses, but I think PTM analysis, especially complex PTM analysis, is a great place to go back to those spectral libraries and collect a paired DDA spectral library. It just makes analyzing this data so much easier because you've got like this huge blue highlight on where the peak should be. So as I mentioned, using kind of these tools versus some of the other histone PTM tools that are available out there has really enabled my collaborators to do a lot more with their epigenetic analyses. So I have a collaborator who's been working on epigenetic mechanisms of neuroblastoma. So one of the drugs that you use to treat neuroblastoma is this drug lorlatinib. It's an alpha kinase inhibitor, but some patients sadly relapse after getting lorlatinib treatment because they become lorlatinib, their tumors become lorlatinib resistant. So this type of mechanism, the collaborators suspect is an epigenetically driven mechanism where the tumor acquires this lorlatinib resistance. So now we can really quickly analyze this data. Now that we've got our Skyline document built, we've got this method to kind of plug and play analyze their data. We just pull that into the Skyline document. We can build even for their specific system an index retention time calculator specific to their preps in their system. I have another collaborator who was working in slime mold. So this is just a video that they have of the slime mold kind of like doing its slime mold thing. It's kind of cool and gross. But the slime mold goes from unicellular to multicellular. So that's obviously an epigenetic mechanism, right? So slime mold is obviously a non-model organism. And we're able to really quickly look at some of these different life stages in slime mold and assess the histone PTMs, the epigenetic marks, as slime mold goes from a unicellular to a multicellular organism. And it wasn't a huge surprise. The chromatin is more closed when it's unicellular and then more open in multicellular. And then I have a third collaborator who's doing epigenetics of ant socialization. So these are leafcutter ants, and they are clonal. So all the ants of a given ant colony share the exact same DNA, but you can see just phenotypically the size is totally different. Their roles are very different. And what he's been doing, he's been asking in leafcutter ants, yet another non-model organism, what is the difference in chromatin marks between these different casts of ants? So this has been a really fun project to kind of push some of the limits of doing good quantification in histone PTMs and PTM work that has really been enabled using all of the tools in Skyline and even coming up with some kind of hacks for how we can use Skyline tools in a slightly different way than they might have been intended. But the flexibility of the software really makes it really easy to once you're familiar with it, figure out ways to adapt it to what exactly you need. So I'm coming up kind of on the end, and we'll take more questions, but this is really the take-home message is that proteomes are more than just abundances. So I talked to you about doing good quantitative proteomics to lay a foundation for any of the functional work that you're doing. I told you about some protein turnover work, using DIA, using Skyline to extract the ion chromatograms. I talked to you just now about post-translational modifications. I didn't get a chance to talk to you about some of my work in protein localization. If you're interested, we can drop some papers. And I'm really excited next to hear more about doing interactomics using DIA from Isa. Big fan of her work, super pumped for that. So that's everything I wanted to cover today. Big thanks to all of my various labs at all of my various stages of training, and I'm happy to take any more questions that you might have and try to rustle up some of those papers if we didn't get to them all. And big thanks to the TAs for covering a lot of the questions. I saw you guys working hard in the chat. Question from Thomas, did you observe if the proteins that had high light to heavy ratio contained more inert amino acid sequences? That is a super cool idea. I like that. I should go back and reanalyze that. We're actually collecting a new data set for that experiment soon. And that's a great idea. Thanks for that. Methionine, cysteine. Yeah, that's interesting. Yeah, I should look for that. What databases do I suggest for histones? I'm not sure what exactly you mean by databases. I don't use any databases. If you mean database search, like search algorithms, I really like Bionic, which unfortunately is proprietary. But Bionic works really great for multiply modified peptide detection. I think Peaks is the other one, which I believe is also proprietary, unfortunately. If anybody has a good free open source suggestion for multiply modified peptides, please let me know. I'm on the hunt for something that would do a good job with multiply modified peptides. Ahmed, is it possible to target peptides that are not prone to modifications for quantification purposes? Of course. So you may have noticed in the IRT calculator, I'll see if I can grab that slide again real quick. In the IRT calculator that we worked on, we specifically tried to get peptides that are not susceptible to modification. All of the N-terminal amino acids are modified here because we perform 2 rounds of chemical derivatization on our histones. So we perform propionylation at the protein level so that all of the N-terminal protein is labeled and all the lysines that are unmodified get modified. We do the trypsin digestion, so everything should end in an arginine, not a lysine, because we've modified all of our lysines. And then finally, we do another round of the propionylation. So now all the N-terminal peptides are modified as well. So these, we tried to get peptides that don't have internal lysines. Of course, a couple of them do, but we tried to focus on peptides that did not have internal lysines so that they wouldn't be modified. Seb, have I ever used DIA for deuterated samples? I have not, but there is no reason that you couldn't use this workflow here with deuterated. You might have a little bit more of an issue with deuteration with the isotopic envelopes overlapping. So I didn't really talk about this, but kind of the idea that people had was that Silac DIA wouldn't work because typically the light and the heavy, these are isotopic distributions. And the 2 boxes represent 2 DIA windows, very narrow DIA windows. Ideally, you would see something like this where the 2 peptides, the labeled and the unlabeled peptide, would fall entirely in 2 different windows. But the concern was if you split the isotopic envelope of a particular peptide so that it split across 2 windows, now you were going to get kind of weird chromatogram shapes. So you were either going to get, it looks like half the abundance that it really truly should be, or you're going to get kind of sawtooth shapes as the peptide falls in and out of staggered window resines. So I haven't tried deuterated. You can try and see if you observe something like this or not. Really easy to do kind of those mixture proteome and check the ratio. That's like a pretty good benchmark for that, I think. Raja, it appears that spectral library is a must in analyzing PTMs. That's not true. So that's true for me in this particular instance. But for doing things like phospho, there are excellent phospho repositories out there. And there's several algorithms that you can use to analyze PTMs in DIA data. So Brian Searle, part of the encyclopedia software suite, is an algorithm called Thesaurus. And he used it to analyze phospho DIA data. We'll see if we can get you that paper. It was NatComs, I want to say like 2018 or 2019. NatComs, it was with Judith Vienne and Mike McCoss, but Thesaurus can do that. I know Spectronaut can do at least single PTMs. DIANN, Diane, can do PTMs. So yeah, definitely you don't have to have a paired spectral library for multiply modified peptides. I think it's just a problem that's a little bit too hard for DIA right now. So right now, I like continuing to do those DDA spectral libraries. Is fractionation recommended for DIA? What type of gradient is recommended? People have been pushing the gradient length for DIA very short. So there's a lot of papers out there right now doing as short as like 5-minute plasma gradients with DIA or swath. So you can definitely do a short gradient. Fractionation, as far as physical fractionation, is not recommended for DIA, at least by Brian Searle and I, at least. We recommend gas phase fractionation. So you can go back and listen to his lecture, and he touches more on why we do not do physical fractionation for DIA. Thanks for getting my back, Thomas. He's got a good reference for the multiply modified histones. A question about the optimal number of gas phase fractions. We did 6. Yeah, if you go back and you listen to Brian's lecture, I think he did a little bit more elaboration on this. 6 was just chosen because we like to do a range. I can move my slides off probably, huh? We like to do a range. There we go. Of 400 to 1,000 MZ. So when we do 400 to 1,000 MZ, it just, the math works out very nicely to do 100 MZ fractions. And that 100 MZ fraction means you end up with 6. Let's see, what else? Concerning isotopic effects, are there preferences for deuterated or C13 labeling? For me and for those simulations that I showed you, that was all using 13C 15N. So that's like as wide a shift as you can get with SILAC. So as heavy as it gets. When you're going down and becoming and decreasing the space between those isotopic shifts, I suspect you might get more and more concerned with splitting that isotopic envelope like that. But again, I would say try it, empirically try it. I love those Navarro style LFQ bench hybrid proteome style experiments. Do a couple ratios of light to heavy and see if they match what you would expect. I think it's probably the best way to convince yourself if it's working or not. How do you examine the data? Suzanne says, to make sure pulse SILAC forms fall in the same DIA window or in separate windows and not in between when you do not get a jagged profile. So you can do simulations like I showed you. If you look in my GitHub from that paper, I have the script that I use to generate those. You can just kind of type in whatever peptide you want to check and see how it would fall, because you could just calculate that, right? You can calculate what the mass to charge, what the isotope distribution should be theoretically for a peptide sequence, and then calculate what the shift would be if you added the heavy isotope amino acid. So you can do that. I think, again, I think the best way to convince yourself, though, like in theory, that's nice, right? Like theoretically, it's nice to see if that is happening or not. But I think it's even better just empirically to prove to yourself, like, is the ratio not what I expect? If the ratio is not what you expect, you might be seeing some of that isotope splitting affecting your quantification, because it's affecting the transmission of ions through that precursor window in your DIA. But yeah, I use those for everything. I use those benchmarking style hybrid proteome stuff for everything. I love them. Okay, so I think we got all of the questions. Oh, question, when would you use encyclopedia over Skyline? Skyline I always use if I'm doing kind of a targeted analysis. So commonly my workflow these days is I'll do my peak detections and my retention time alignment using encyclopedia. That generates a library. You can straight up generate a blib library using encyclopedia, and then I put that in Skyline, and then I can extract to view and manipulate my chromatograms in Skyline. So if you look at that paper that Brian had mentioned, the MCP paper from 2022 that Brian and I put out, the supplement number 1 is a tutorial that walks through like step by step exactly what we do, integrating encyclopedia with Skyline for our analyses. Do you think DIA could be used for something like thermal proteome profiling? Absolutely. It's all the exact same thing, right? So that's kind of like you're just measuring peptides and then doing some post-processing. So I don't think there's any reason you couldn't do it for thermal proteome profiling. Nice. So we got all the questions.
Well, this is fantastic. Thank you so very much, Lindsay, for the excellent presentation. I am really amazed by